right, so welcome to Chapter 1 of CLLS 5319. This isn't in the textbook, but I wanted to just give you a quick overview, and then we'll start with Chapter 1. What is biostatistics? So it's the application of statistical principles to medical, public health, and biological applications. You collect, summarize, interpret information, and make inferences that appropriately count for uncertainty. What are the different types of studies? We've got laboratory studies, animal studies, clinical studies, experimental trials, and observational studies. Research teams will consist of the principal investigator, a biostatistician, co-investigators, project manager, statistical programmers, and research assistants. They don't always consist of all of those people, but it can. The biostatistician's role on the team is to come up with a study design, or to help with the study design, the research question, figure out who the sample should be and how large it should be, enroll the people in the sample, and try to keep them in the study for as long as possible, follow-up strategies if necessary, and to provide ongoing monitoring. They do the interim and final analysis, and they interpret and report results. Okay, so in Chapter 1, we're talking about statistical and critical thinking. Specifically in Chapter 1.1, we're talking about sample data and how it's important to consider the context, source, and sampling method. We'll talk about statistical significance versus practical significance, and voluntary response sample and implications for interpretation. So, when you're looking at a sample, it's important to consider how the sample was formed. Was it through random sampling? If not, findings can be suspect. Not in the way that they're not true, but they may not be representative of the entire population. Statistical significance occurs when you find a result that was very unlikely to occur because of mere chance. But even though you may have statistical significance, you may not have practical significance, because even though you may find a statistically significant result, it may not be practical to pursue that line of research or that particular intervention. So if you'd like to read more about that, see page 8, example 3. When samples self-select to participate in a study, the findings from these studies can not be as persuasive as findings where the sample were randomly selected. So for example, if you're doing a survey study and you solicit responses on a conservative website about the performance of a Republican president, the findings that you get from that particular sample will skew conservative. It won't be representative of the overall population. Similarly, if you survey about a smoking cessation drug in a health magazine, people who choose to take that survey, I should say, are more likely to be on the drug. So what are the different types of data? We're going to talk about a parameter and a statistic. We'll talk about quantitative data and categorical data, discrete versus continuous, and whether basic statistical calculations are appropriate for a data set. So, all right. Why do you have to take this course? Why do we do statistics at all? If I were to take a measure of a population, right, I wanted to know how is the whole entire United States going to vote on the next presidential election, for example. Well, we would just call that the election, right? But we have these things called polls, and we draw samples of maybe 1,000, several thousand people, ask them who they're going to vote for president, or ask them some health-related question. And then we take the results of that sample and generalize the findings back to the population. Why do we do this? Because it's much more doable to take measures of a smaller sample than it is to do an entire population. We're in a census year this year, and we're trying to just count how many people are in the United States. That alone is difficult enough. To actually get a measure of the entire population is almost impossible. So we use a sample, and when we do that, we call them statistics, which I'm about to talk about. All right, so most everybody knows that in statistics you're going to see some Greek letters, and it's not something that you need to freak out about, because I'm going to give you a trick to remember. When we talk about a measure, a numerical characteristic of a population, it's called a parameter. Now I have the P underlined in red, and I have the parameter, and I have the P in population. Luckily, they both start with the letter P. We're familiar with the Greek letter pi, which just is P in Greek. So when you see Greek letters, we're talking about a measure of a population, which is called a parameter. Okay, so parameter P is a numerical characteristic of a population. It could be an average, it could be a median, it could be a mode, whatever. If it's written in a Greek letter, we're talking about the population, it's a parameter. All right, down at the bottom, that funky looking backwards Y is called a mu, and that's the mean. Sigma squared is the variance, and sigma is standard deviation. You don't need to remember that right now. I'm just giving you examples of Greek letters that represent parameters of a population. So when you see no Greek, we use the alphabet we're used to, then we're talking about a statistic, which is a descriptive measure of a sample. Interestingly, they both start with S. So statistic is to sample as parameter is to population, okay? And when we have Latin alphabet, the ones we use every day, the letters we use every day, then you're talking about a statistic, which is a measure of a sample. So down at the bottom, you'll see this X with a line over it. That's the mean or average. It's pronounced X bar. We're going to use just the letter M. I'll say that later on in the slides. S squared is the variance of the sample, whereas sigma squared is the variance of the population. And S is a standard deviation of a sample, and sigma is the standard deviation of a population. Okay, so we have different kinds of numbers, right? So when we're talking about quantitative, quantify, we are talking about numbers that represent counts or measurements. For example, weight, height, blood pressure. But we also have this thing called categorical data, which is also called nominal data. See the Latin root nom in the word nominal, which means name, or if you speak Spanish, nombre means name. So nominal is a naming variable. These are names or labels that we use. We use numbers to code data. So 1, for example, could be male and 2 is female. But these represent categories, not magnitude. So just because I say male is equal to 1 and female is equal to 2, it doesn't mean there's more or something better than in terms of female because there's twice, it's just a label. It's just a way that we tell the computer to count it up, but it doesn't mean quantity there. Okay, it'll become clearer as we go through. All right, so it's called the levels or scales of measurement. If you speak French, noir is the word for black. And if you've heard of film noir, there's a picture from, I think, The Postman Always Rings Twice. But the word noir also is an acronym for the different levels of measurement, which is why I'm bringing this up at all. So we have nominal, ordinal, interval, and ratio. Nominal, I have the line pointing to the male and female, like I just said. These are also called categorical, right? So nominal or categorical, and male and female are just labels, right? Ordinal, I have the line drawn to birth order, right? So first grade, second grade, third grade, fourth grade will go in an order, right? One star on a Yelp review, 2 stars, 3 stars, 4 stars. The difference between ordinal and other numbers is if I give a restaurant a 1-star review and you give a restaurant a 2-star review, we can tell that you liked it better, but we don't know what your 2 stars represent compared to my 1 star. We may have liked it the same. I just gave it a lesser rating. The distance between a 1-star and a 2-star and a 3-star is not necessarily always the same. So we call that ordinal data. Then we have interval data, and I don't know if you can really tell, but that's a picture of a thermometer. And the reason why I have it pointing there is when you're talking about temperature, if I say it's 0 degrees outside, it doesn't mean there's no temperature. It just is a placeholder in the order of the thermometer, right? So interval data, each line between each degree on the thermometer is equal distance, but 0 doesn't mean absence of, it's just a placeholder. Whereas ratio, I couldn't make the line go through those pictures, but ratio is that bathroom scale over on the right-hand side. And that, when you get off the scale, it goes to 0 because there is no weight on the scale. And then when you get on it, it tells you your weight. So we have nominal, ordinal, interval, and ratio. And those are the scales of measurement, or the levels of measurement. And here's just a chart that says the same thing. So we can talk about nominal, eye color. I'm looking at the examples. Ordinal, ranks of colleges in the U.S. News and World Report. Interval, body temperatures in degrees Fahrenheit or Celsius. And ratio, heights, lengths, distance, volume. All right. So we have discrete data. A set of data is said to be discrete if the values belonging to the set are distinct and separate. They're unconnected. So number of births and deaths, you can't have a half of a live birth or a half of a death, right? It's a whole number. Number of patients seen, you can't see half a patient. Number of people in this class, etc. Okay, that's discrete. Continuous data is a set of data that's said to be continuous if the values belonging to the set can take on any value within a finite or infinite interval. So the height of a patient, blood pressure, temperature, right? You can have fractional spaces in between the height of me and the height of you, right? Big data and missing data, extreme. So now, especially, with the computing power that we have, we can have extremely large data sets where the analysis requires advanced computer capabilities, and we call this big data. This is kind of the marriage of statistics, computer science, software engineering in the context of relevant fields such as epidemiology or biology, which is very, very, very relevant in the current day and age. Missing data can become very complex about how to handle it, but in brief, what's most important is why data are missing in the first place and what would impact the interpretation of the results. Because if data are missing at random, removing the incomplete observations doesn't really necessarily impact the overall findings, but if data aren't missing at random, then if there's some underlying reason why they are missing, it will impact the findings. For example, if you're giving a survey and you ask somebody about their income and people skip that question, right? Some people do and some people don't. It's possible that the people who skip that question maybe are low income and don't want to report their actual income. So that would be a case where it's not missing at random, and then you're missing out on all of the people who don't answer that question. All right, so collecting sample data. We're going to talk about simple random samples and talk about the importance of sound sampling techniques and good design of experiments. All right, so we have the gold standard, which is the RCT, the randomized controlled trials. It's called the gold standard because they are the most effective. You can have experiments where you apply some treatment and observe the effect on the individuals, which we call subjects in our studies. Or you can do observational studies, observe and measure specific characteristics, but you don't attempt to modify the subjects. For example, it would be unethical to assign someone to a smoking group and a control group, right? We know smoking is harmful to people, so we wouldn't want to inflict harm on our participants in our study. But we could do an observational study by asking people, how many years have you smoked? Okay, then you represent our smoking group, right? They self-selected, they chose to smoke. We're not asking them to go do that, but we can actually compare those people who smoked in the past or who are currently smoking to those who are not. All right, so when we design experiments, it's important to see if the effect is the same with other samples and other settings. So we want to do more than one study. You do one study and you find a finding, that's great. You need to have replication in order to believe that it's true. When we do blinding, it's important that the subjects and experimenters, so if the subjects don't know, it's a blind experiment, but if the subjects and the experimenters don't know who's getting the treatment, who's getting the control, it's called a double blind, and they either receive the treatment or they get the placebo. If you find out, if you don't know, if you think you're getting the treatment, you can have a placebo effect where even though you're getting the placebo, you see some sort of an effect because they feel like they're getting a treatment. It's just a phenomenon that has happened. Randomization, it uses chance as a way to equate groups when assigning subjects to the treatment or control. This is the only way that we can actually, in any way, know that the groups that we have selected or we have assigned to treatment and control are the same, is if we use randomization. Otherwise, you're not going to get it. And it's important that our groups are as equivalent as possible before we administer a treatment or control, because if they aren't, then we can't be sure that the effect is due to the treatment or if it's just some other confounding effect. Okay, so simple random sample is the easiest way to do. You just names out of a hat, or a computer randomizes subjects to treatment and control group. Specifically, every possible sample of the same size, which when we talk about sample size, we say n is the sample size, has the same chance of being chosen. There are other types of sampling. Systematic, every case person is chosen. Convenience, stratified, cluster. All right, I'm going to go in, the next slide has the picture, so I'll talk about that. So we have simple random sample up at the top. A sample of n subjects is selected so that every sample of the same size n has the same chance of being selected. A systematic sample, this implies that you have a list or some sort of sampling frame where you have all of the potential subjects from which you can select. So you initially pull maybe a number out of a hat and say you pick the number 3, so every third subject is selected until you get to the size of the sample that you want. A convenient sample is data that are easy to get. There's a thing called snowball sampling. If you ask one person to refer you to the next person and that person refers you to another person, it's like the snowball rolling down the hill. As it goes down the hill, it gets bigger and bigger. That's called snowball sampling. It's also a type of convenience sample. If you have access to people in your classroom and you want to do a survey, you could do a convenience sample of people for whom you have access. Then stratified sample, if you know that the population that you want to sample from skews more female than male, say for example nursing students, maybe there are 60% females and 40% males, you would want to pull your sample to represent 60% female, 40% male. And that way you're as close to the overall population in your sample as possible. And then a cluster sample is when you partition the population in clusters, then within those clusters you randomly select. Then select all the members of the selected cluster. Okay. So for example, if you're looking at this square here, you can't tell what's behind there, right? We want the sample from the population to describe the picture, the big picture. If we randomly sample enough of the population, we can see a rough picture of the population, but this sample isn't big enough. I can't tell what's going on there. If I pull enough people, I can see an outline of a face, and if my sample is really big, I can tell that that's a famous photograph of Einstein with his tongue out. But if we select individuals versus clusters, we may, so the one on the left represents individuals, a very large sample of individuals, sampled from the population. If we do clusters, we sometimes can miss out on what we're trying to describe. And if we have exclusions from the population, we can sometimes miss out on very important information, like whether or not his mouth is open, whether or not he's wearing a tie, whether or not his eyes are open, for example. What kind of exclusions? Maybe we don't have access to people 65 and older, so we just blow them off and say, you know what, we just won't sample those guys. Well, that's okay, but you may miss out on the big picture. And remember, the whole point of this is that we can't go ask everyone in the population some question, so we use a smaller sample to represent the population. And if our sample is somehow substandard or somehow not representative, then we're not going to get a big picture of the population. All right, so we have different types of experiments. We have cross-sectional, data are observed, measured, and collected at one point in time. We have retrospective or case-controlled studies. Data are collected from a past time period by going back in time. All right, like a smoking study, you know, how many years have you smoked? All right, then we'll put you in that group. But you're looking back in time, and you're not administering some sort of intervention or drug or treatment. It's something that has occurred over the past. And then you have prospective or longitudinal or cohort studies. These are data collected into future from groups called cohorts that share a common factor, like those at risk for myocardial infarction. All right, observational study, observe and measure but do not modify. When are the observations made? In the past, it's a retrospective or case control. One point in time, it's a cross-sectional study. Forward in time, a prospective or longitudinal cohort study. Okay, confounding. I said this before. When there's an obvious effect, but we can't quite identify the specific cause. This can be alleviated by using the following designs. You could have a completely randomized experimental design. You use random selection and random assignment to treatment or control groups. You can have randomized blocks design, form blocks of subjects with similar characteristics, typically gender or age group, randomly assigned to treatment or control within each block. And then you could do a matched pairs design. This is very good for a before and after, measurements before and after some sort of treatment, or you could do a twin study or a spousal study, depends. If they match on some sort of, there are individuals who are somehow connected. They live in the same house, they are, you know, identical twins, you can match on that. If it's group A at the beginning of the semester, like you guys, if it's you at the beginning of the semester and then I test you again at the end of the semester, that's the before and after design. You're the control group at the beginning and then you're the treatment group at the end. Sampling errors. So sampling error or random sampling error is when there is a discrepancy between the sample result and the true population result. Non-sampling error is the result of human error. And non-random sampling error is not using a random sampling method, right? So convenience sample is definitely a non-random sampling method. So you would have non-random sampling error in that. All right, so we're at the end of the chapter. Please do the practice problems, and the quiz will come directly similar to the practice problems.